Okay, well now we're moving into the fourth and final major category or area that we could subdivide classical genetics into. We've looked at transmission genetics, we've looked at molecular genetics and learned about expression of genes, mutation of genes, regulation of genes. We talked about chromosomes and chromosome variation, and now the rose among thorns, if you will. And I say that because it's the area that I've had my training in, primary training in, and have spent most of my career working in. This is population genetics. So what I'd like to do in a series of lectures now is to talk a bit about what is population genetics. In this case, why is it important? And we'll try to illustrate that in our first part here. And then we'll come back and really dig into the elements of population genetics. This first part, I'm going to give us a basic definition. We'll come back and actually give another definition later. But this kind of points out all the elements of it. And then I'm going to come and give a few examples of where population genetics plays a role in helping us understand our world around us, human systems, animal systems, plant systems, and why it is actually important to us and where we work with. So what is population genetics? Well, as this says, and I took this right from the journal Nature, but and it's a good definition. It covers everything, so we'll just start with it. And again, I'll give another one later. But population genetics is a sub area of genetics. It's a study of genetic composition of populations. That's kind of circular definition, but it means that we're studying what's going on in groups of organisms. So we're not just interested in a single cross where we've got A times B gives me C, and I want to predict some outcome of that. I've got lots of A's times B's everywhere, lots of C's being produced, and I want to know, as a collective, what's going on. So what I have to look at are distributions of both genotypes and phenotypes, and their frequencies of those distributions. So we're going to talk a lot about frequencies. And what we're really interested in here are changes, how things change in terms of the genetic composition, what's going on relative to the genotypes and the phenotypes. And we know from study that there are a number of processes that can be involved. Amongst those are natural selection, so be choosing the tall ones versus the short ones, or the short ones versus the tall ones. We can have what's known as genetic drift, which is chance events are going on. We've studied mutation ourselves. Well, we're going to talk about what the effect of mutation is on a population. And then gene flow, what happens when individuals get up and move around, or we send our propagules out into the water column if we're a fish or something. So we're going to talk about all of those processes in terms of what's happening to this genetic underpinning of populations, of groups of organisms. Now, where did all this come from? Well, once again, we go back to Darwin, right? This is really the start. In his Origin of Species, he set down the ideas about natural selection. And we've talked about how natural selection might take us to giraffes, or might take us to all sorts of organisms by making them fit, providing them with some genetic compositions that would allow them to somehow be better than some other organisms or some other variations of that same organism. And Darwin was interested, as obviously. He was also, of course, interested in the genetics of all this with his pangenesis and the ideas that he never really liked. But he brought forth these ideas. And we're going to talk about selection. Now, actually, he not only talked about selection, but many of us are trained as biologists and things that selection is the big force and that's driving the systems, and it certainly plays a major role. But even Darwin recognized there were other elements involved, and one of them, in fact, is randomness, is genetic drift, as we call it today. He said that those traits that weren't under selective pressure were just randomly changing, and that becomes a very important thing. It's something that a lot of people miss when they talk about Darwin and Darwinism. Another thing that people miss is that Darwin never mentioned in his book the word evolution. He never said that in The Origin of Species. He did use the word evolve one time, but he never talked about evolution. He certainly didn't talk about humans coming from other creatures or something, but that was obviously the implications that we arise from pre-existing things. It wasn't until many editions later that the editors actually put the word evolution in the Origin of Species text. So if you go back and read the first few volumes of it, which are really his words, without all the extra editing, it doesn't even appear. Now, again, he was very concerned with this idea of pangenesis. He thought it was not sort of a bad idea, didn't make much sense, it didn't work well. It was conflict with other models of evolution. And so he spent the rest of his career looking at plants and animals and things and trying to understand this whole process of genetics without understanding that Mendel was right out there. And eventually he did get forced to talk about the descent of man and how humans had come about from this long lineage of one organism leading to another. But actually, in that text, probably the more important thing for most biologists isn't this descent of man, but this idea of sexual selection. He was really looking into this idea as well of why do certain things just go extremely, off the beam on size or color or all these other things. What is sex doing at selecting those traits? This is a widow bird here, and the widow bird has such a long tail it can barely fly. How in the world is that an adaptive advantage? What's going on there? Well, maybe it's attracted to one of the sexes, and therefore, the ones with the longer sort of runaway selection. And that's still being tested today, and it's a very large component of evolutionary biology. All right, so we have Darwin. Okay, now obviously when Darwin's book came out, there was a huge uproars in many directions. We all know about the ones based on theology and ideas there, which still persist today in many cultures, including in our own. The ideas of evolution and how that plays a role, and etc. There was also controversy in the ideas of what was called social Darwinism, which because we were in the industrial age and we had these great people of great wealth, the robber barons and people, and they said, well, look, we're supposed to be wealthy. Our families should be wealthy and we should have all the power because we're selected for that. We're not, and actually that seems to carry a little bit on today with certain families of the world thinking they deserve to be the most powerful. But that was called social Darwinism. But for us as biologists, what we're more interested in is what biologists were fighting about, and they were fighting. There were two camps that came about. One was the sports camp, this idea that when you started to domesticate something or you started breeding animals, you would see these things called sports. Okay, I've got them in quotes. Sports were mutants or deviants that would appear, right? We'd see something that's suddenly albino or something with no tail or whatever. And this is in the 1800s. This is a time when in the UK, there a lot of our dog breeds came about. People were just frantically breeding dogs and getting all kinds of new varietals, new types, and then chickens and everything, pigeons, all sorts of things were being bred. And these, when you start doing that, start inbreeding, you start seeing these sports. Well, this led to what was known as mutation theory or the idea of discontinuous evolution. On the other side, people said, no, look, evolution doesn't just make these huge jumps. It does it very slowly and it's kind of process. That's what Darwin said. Well, Darwin was kind of in the middle because he was studying sports the rest of his life, and he certainly had put forth the idea of slow, continuous evolution. The people studying continuous evolution eventually started looking at like distributions of size and things, this classic model here. But you can think about it. If you go into any room and look around, and there's large down to small, tall to short, right? We don't tend to be completely all tall and all short or something else. Interestingly, these led to a group of individuals known as the biometricians. And they were interested in looking at things like what's called mid-parent, where they would take the height of the parent and the height of one parent and the height of the other, and they would put them together and see and predict that the offspring would be somewhere in the middle. This whole idea, again, of blending inheritance almost, right? We blend back to it, and that was, of course, a big problem for Darwin. But they actually, that whole method of comparing those, where you take one line, y, x and a y, and you run a graph, is regression. The word regression actually came from regressing back to the middle, right? So it's coming after this sort of Darwinian ideas. Well, anyway, eventually the biometricians developed into what we think of today as statisticians. And this is where a lot of our statistics came from, was the individuals actually trying to solve this problem of Darwinian natural selection and things. Now, of course, the French had been for long working on probability theory, which fit right into this, but that's the other major part of statistics. But a lot of what we know, ANOVAs and regressions and all these basic models, chi-squares and things, came from individuals that were working with genetics, trying to figure out this natural selection. Well, on the other side, eventually we got down to being what were called Mendelians. Mendelians were individuals, once Mendel was rediscovered, uh, realized that this discontinuous idea might work, right? You did have dominant and recessive and all these sorts of things. You had the separation. And these groups fought. I mean, they fought and fought and fought. It was very, very nasty. The biometricians would be, someone would become editors of a journal and they wouldn't let anybody that was from the other side publish and vice versa. That was quite a really sort of nasty scientific fights that went on about what was going on because it's critically important to understand, right? Well, of course, in 1900, we had to read, Mendel was rediscovered, and we'll talk more about that later. And Mendelian ideas came to the forefront. In about 1908, we're going to see that a very important event occurred that kind of helped move the Mendelians forward. But actually, it wasn't until 1918 that these two groups could have coalesced. And they coalesced under this individual who was named R.A. Fisher, who wrote a seminal paper that came out in 1918 explaining how these two things link together, how you could have all of this work under one model. But notice that's 20 years after the rediscovery of Mendel, almost 20 years, and it's about 50 years, 60 years past when Darwin first published his. Okay, but what Fisher did, R.A. Fisher, was start the whole idea of population genetics. Very brilliant man. ANOVAs, the idea of ANOVAs were developed by Fisher working on agricultural stations when he was looking at how to plant crops and get the most information from these tests. The F test, if those of you who've done statistics, is for Fisher, right? So he did lots of amazing work with statistics as well as theories of population genetics. When this came about, and starting in about the 1920s, Haldane was someone that was just important. He worked on selection himself, but he worked primarily with selection in humans and human ideas. He was British, very famous British developer of this whole area of population genetics. And then we, in the United States, we had Sewall Wright. Sewall Wright was a professor, and he worked a lot with, particularly with looking at this idea of drift, the idea that you could have these, you had selection working, but you also had drift. And he came up with this whole idea of these landscapes where selection would push you up toward the top. Once you got there, you couldn't move. And the only way you could move to another peak, even a higher peak, was for drift to shake the whole surface around. It's wonderful theories that he was contributed to. But these three men and their writings initially started this whole field of population genetics. Now, what is it today? Well, it's all sorts of things. You may have heard of geographic medicine, that which looks at where diseases are, how they spread, forms of those. Those are some things. Conservation genetics, some things we'll talk about. The idea of, all right, how do we preserve these populations, and where humans are really building and developing, and of course with things like climate change that can happen, you get populations isolated and separated from their original ranges, and so how do we maintain those systems? And it can be all sorts of ideas about how that works. Let's talk about some of that. Getting back to humans, genetic counseling is really just population genetics for the most part, with molecular genetics nowadays thrown in, but it started as population genetics. Just what's the probability? I need to know the frequency of this mutation in the human population. And given that frequency or occurrence, here's your probability of you two having a child with or without the disease, and that sort of thing. Forensic genetics is classically there. You played a little bit with that in labs, where you talked about maybe a murder or something. Well, if you're going to argue that this marker you have, this molecular marker, says this person is guilty, well, what's the frequency of this marker in the population? How many other people in town could have had that particular marker? Whether it's ABO blood group, going back all the way to that, to modern fancy things that we have that can detect, 1 in a billion occurrences. So forensic genetics is there. Ecological genetics is another area where we just look at populations in general and how they persist and change and exist over time, and one of the major areas that we work with. And then that all leads, of course, up to evolutionary genetics. What's happened over time? So what has happened over the last million years or 2 million years to change and shape these things? Why do giraffes look like giraffes, right? That's evolutionary genetics in its broadest sense. All of these, at the core of them, have one thing, which are called allele frequencies, right? Once you get a group, you just don't have an allele, you have frequencies of alleles. They're found in 20% of the individuals, 90% of the individuals. What's the frequency? That becomes the base unit. That's our currency which all of our models are actually built on. Okay, so given all that, let's take a few little examples here. Here's one. Um, with humans, we're really interested in the idea of where humans, at least Homo sapiens, came, and there's almost daily there's some new information out. We've pretty much come to the idea that we are of African origin, and that we moved out from there, and that's for population genetics, looking at the frequency of alleles in various populations around the world and seeing how they relate back. With one idea being that where the most genetic variation occurs is the area probably of original origin, and the most variation in humans in one area is in Africa, which is consistent with the fact that hominids originated in Africa and then spread out. And some more recent data information on this is about how often the Neanderthals came out and Homo erectus, and that there was clear inbreeding between Homo sapiens, us, and Neanderthals, because people still carry Neanderthal blood around, right? Or Neanderthal genes around, I should say. Okay, but anyway, that's population genetics, how that works and where it comes from. And then when you start tracking humans, you can also start tracking other things like uh, head lice. The genetic variation found in the forms that are found in head lice tend to track where humans have been. And there's more varietals and more genetic variation in head lice in Africa than there are in other parts of the world. And it helps us kind of understand the patterns that have occurred over time. And it's not unusual to say, wow, head lice is kind of weird. Well, all sorts of mammals and other things have parasites, right? And there's a synergism between the types of parasites and the organism. And you can actually sometimes they're so specific that you can look at the parasite and tell what species they're on. They're specific to one species or so. So they co-evolve. It's called coevolution. A really interesting area to study. Taking that even further, there's some theories about the coevolution of the genomes of those two individuals. Once this individual becomes parasitic or symbiotic or whatever, parasitic on top of an organism or in an organism, its genes start to modify to mesh with compatibility of that host. And so there's this whole relationship between host and parasite or host and other forms genetics. Very interesting. And then with humans, we've had things like leprosy, been around for a long time. Leprosy kind of showed up, and people were wondering where in the heck, where did this come from, right? Where did this disease arise? And some studies that were done a few years ago started looking at strains. They were able to go in and pull up medieval bones, and actually from those bones find that they were obviously had the evidence of leprosy in them, and they were able to extract DNA from those sequences. And when they did, they went back, like you built some trees and things, and started building trees and figuring out where the systems came from. And it looks like in New Caledonia and China, we go back almost 1,000 years before, so 3,000 years ago. Okay, a lot of it, the variation goes back about 2,000 years. So we've had leprosy around in populations for a long time. What's really interesting about all this is if we track all of this variation back, clearly where leprosy came from was the Americas. So the Americas brought this leprosy to the rest of the world in some forms, which is kind of, I think, incredibly interesting idea. All right. So, and this is population genetics. This is what we do. We look at the frequency of alleles, look at frequency of variation of mutations nowadays with using DNA like you've done in lab, and we track back and we build these topologies to help us understand where these variations, how they all fit back together. One other really interesting one, the reason I picked up this little armadillo here, the 9-banded armadillo, Davis novicinctus, is only one of the only other mammals that has leprosy. And they're actually used as a study model for leprosy. And it turns out that humans gave the strain to these guys. Okay, armadillos didn't provide humans with the leprosy, but we actually gave it to them. That turns out to be an interesting outcome. But lots of other uses, and we'll just kind of flick through a few. We look at what's called admixture in multiple populations. Many of you may have taken those little DNA tests. I've talked about that before, but where you get back and you find out, yeah, from 10% of Scandinavian and 42% German and 10% Native American, or a 4% is Jewish or something. Well, that's population genetics. That's all that is. Those frequencies and understanding those relationships. And it's not just humans. We work with things like trying in conservation biology, trying to understand, okay, we only have a few real existing populations of some animals left. At one time, they had these massive ranges like these giraffes did, and same thing with lions and tigers. Lions used to spread all over Eurasia, and now they're down to these very small groups. We're trying to conserve them and understand them, and we've got these various forms. Well, it turns out now that even though we got all these various forms, we thought they were one species, but very recently, in the last year or so, they decided actually no, these represent 4 functional species groups of giraffes. So if you're managing giraffes, you don't want to take some from up here and just randomly throw them in down here, because you're actually interacting with species differences, not just population level differences. Okay. I've worked on a number of things over time. Had the pleasure of working with some individuals many years ago on Onchocercus, which is the African river blindness. It's a worm that gets in the body through a bite of a simul fly, a black fly, and it's from Africa. And it turns out there are 2 forms in Africa. There's a form that's where it's a host is an ungulate, so it's in the plain, so it's like it might be something zebras or gazelles or something. Uh, that would be an ungulate form. And then up in the sort of equatorial part of Africa, the form that's there is a simian form. It's carried in monkeys, great apes, and things. And it turns out that you treat these slightly differently. They respond different to medications. Both of those have ended up in the New World because of the slave trade. And we were working down near the Amazon and stuff. I was only working on the data, unfortunately. I didn't get to go to the Amazon. But there, it turns out that these worms are actually hybrids. There actually turns out from our data that they're a mix of this ungulate form and this simian form that occur there. It's kind of fun. Stuff I did at other times too. We would try to look in Indonesia. I was in one project where we were looking at trade, and trade there was pottery primarily. Individuals would move pottery around, and then language would move, with the trade routes, right? That, and so the question was there gene flow or not? Was it just, were the trade routes just trade routes, or was there not? And the linguistics ideas. So we were able to do these 3-way comparisons where we looked at gene flow relative to the pottery models that were out there. So what type of pottery were found in different areas historically, and it was this historical data versus the linguistics type ideas. So we looked at how gene flow related to both of those and came up with a nice model looking at that. It's kind of fun. Again, I didn't get to go to Indonesia either. I got to work on. Sometimes we just work in the behind a computer. A couple of little studies we did. One really fun. We were given data from some grave sites, some 8th century graves, what's now Hungary. And there was a big fight going on at the time about between some anthropologists about what the pattern was in these grave sites. And one of them was that the idea was that there had been an invasion. Some peoples had come in, they had assumed the properties of others and had made the others servants to them, right? So they had the people that were there originally had become servants of this overclass that had come in and taken over. And based on some of the artifacts and some of these grave sites, it was proposed that there was actually a kind of a movement, they assumed together, so that the people that were there originally, the natives, and then the people that came in and took over kind of just meshed families eventually. And so they were buried sort of as these groups of individuals. And other people said, no, that's not what happened. What happened is they were never really assumed in. They were treated strictly as probably as slaves, but treated differently. They were not part of the family units, and they most certainly weren't buried together. So we got some ABO blood sent to us from ABO blood samples, and we got to test these 2 hypotheses based on the frequency of the ABO alleles in these populations. It was really a lot of fun. So this is called the crescent moon hypothesis, the idea that the individuals that were there, the native individuals, were never assumed versus this one where they were brought in. And it turned out that this crescent moon won out, even though some of the physical artifacts looked like they might have been combined in like this. The genetics was pretty clear. They were very separate groups and they hadn't meshed together very well. And just one little last thing, other studies, just if you're interested, black and white crappie, and they're in the impoundments in the South, and people would go out and they hybridize, and there were all these ideas that they would only hybridize in one way. It was always a black female with a with a white male, or a white male. It was all one way or the other. It was always unidirectional, right? And the guides would tell people that no, and they only do this. And so in the fisheries department here, they collected quite a bit of data, and so we went in and looked at mitochondrial data, which comes from females, right? And these are in fishes, and it's like everything else, and compared that to nuclear data that we had showing the mixing, and asked the question: is it one directional gene flow? Is it always a white female with a black male, or a black male female with a white male? Which direction is it? And it turned out to be none of that. It turned out to be essentially the frequencies we found both hybrids. We found both males, both white and black would be the females, and the frequency of the hybridization was purely the frequency of the fish in the population. So if there were 70% black crappie, about 70% of the hybrids were black. From the black side. If it was 70% white, then 70% were white. So it was almost perfect match to the actual capture rate of these two species in the population. So it solved a nice little question for people, and it was a good little project for somebody. And those are just some examples of population genetics. We will hit some more of these as we go through and kind of dig in depth of what's going on. So the next lecture, what we want to talk about is allele frequency. So we're going to set up a model, and then we're going to talk about allele frequencies underneath that model and how those can be used and or at least calculated for our purposes.